Okay, thank you. As I mentioned, we're going to move into now a little bit more life science area. And first of all, I'm a postdoc in Jens Nielsen's group, and we're in the biology department, or biology and biological engineering department. And so I think a good way to demonstrate or give a good example of the type of work that we do and how I think that connects well with the ICT area is going through a specific project that we're working on right now. And that involves looking through human omics data for cancer biomarkers. First, I should probably define or at least clarify what I mean by omics data. When you look at the general flow of information in a biological system, you have a single gene which is encoding an mRNA transcript. The gene is expressed. The mRNA transcript is then translated into a protein, and this is what actually performs the functions or performs the structure of the cell or biological system that you're looking at. And that protein, in addition to functions, one of those may be catalyzing or driving a metabolic reaction. But each one of these is just a small piece in a much larger collection or library spanning the entire cell or biological system. So for example, all the genes in the DNA of the organism form the genome, all the transcripts, mRNA transcripts of the transcriptome, proteins, proteome. I think you get the idea. But what this has done is now that we're actually able to get, with high throughput technology measurement devices, we're able to get snapshots of these entire, or virtually spanning these entire library of molecular data. And in this way, molecular biology has become very much a data-rich discipline, and that's where this connects well with this digitalization concept. And a specific set of omics information that we're interested in is cancer omics. And so one particular database that's been useful to us that holds a lot of this very nice cancer omics data is the Cancer Genome Atlas. And that has data from over, I think now, 10,000 different patients spanning many different parts of the body, different cancers. And we're looking specifically right now at transcriptomic data. So this is gene expression data, the amount of each gene that's present in each of these samples. And again, this spans, in this case, 32 different cancer types, and there's different types of samples. So for example, you can actually get these paired samples, which is very useful. For a patient, you get not only the tissue sample from the tumor, and we get a profile on the gene expression from that sample, but you also get the normal tissue directly next to that tumor. And so you have this nice paired situation, which I'll get to in just a second, so keep that in mind. And we're not interested actually in the entire collection of genes, but we're actually specifically focusing on a set of genes making up the secretome. And so this is, I'm throwing another ohm at you, but eukaryotic cells, so human cells, have this protein secretory pathway, and as the name would imply, it involves in packaging, folding, and secreting proteins. And from this collection of genes, the subset of genes that encode the secretome, why this is of interest to us is that these are more likely to be found in biofluids, so blood and urine, which are able to be sampled in a much less invasive manner than a typical biopsy, where you have risk of infection, in addition to a limit on the frequency. There's only so many pieces of someone's liver you can remove before they start to complain. But looking at the data of the secretome, and we can now do it PanCancer, so comparing across many different cancer types, this just gives a sense of what the data looked like, where each point on the left here is a different patient. And you can see initially just the spread looking at the data directly, and I'm showing a subset of cancers just to help ease of viewing here. It groups very much by tissue of origin, and this makes sense. Obviously, your liver cell is going to express a different set of genes in a different amounts than a cell in your stomach, at least we would hope so. But what we're interested in is actually separation or these patterns that are specific to cancer. And so what we can do is take those paired samples I was talking about and look at the change between normal and tumor transcriptomic profiles. And in doing this, because you're looking at fold change within a patient, within a tissue, a lot of those tissue-specific, patient-specific effects then fall out. And that allows this nice comparison and can give us information that's specific to the cancer. And one direction that we took with this, and there's essentially an infinite ways you can look at this data, is we looked at the extreme changes in these genes for each cancer type. And when we look at genes that had a very high increase or decrease in expression, we organized those into just a matrix here. And we can see actually that the majority of these extreme secretome genes we found are specific to a unique cancer type. And in this sense, they constitute a very nice biomarker unique to that particular cancer. Of course, there's follow-up experimental validation that's required to actually move this forward, but this just gives a sense of the type of information that you can pull from this type of data. Of course, there's a lot of challenges currently with this, these types of data and these approaches. There's a vast amount of variability, not just between different cancer types, but between different patients. Also within the tumor itself, different cells in the same tumor have a lot of heterogeneity and variability. And the directions that we're moving in the future to begin addressing these and other problems is beginning to integrate different layers of data. So not just the gene expression data, but also the protein levels, the metabolomes, so the concentration of metabolites, beginning to integrate those, as well as incorporate these data sets with biological data. So in the form of, for example, a network where you can then map that to a network. And this can be done on the tissue level to become tissue-specific or on the level of a patient. And this actually is part of this drive toward personalized medicine, where you have a treatment or a biomarker that's tailored specifically to your biological profile. And so that's the direction that's moving toward. So with that, of course, I'd like to thank everyone here, as well as my advisor and Jens Nielsen, and many of the people that I work with in my lab. So thank you.
Thank you, Jonathan. Thank you.